Well, it's basically a prize that says when you're a cell, if you're an animal cell, you have to always have some level of oxygen. Almost all cells use it to do their metabolic processes. And basically, it is just like a candle burning or any other kind of furnace or engine. You are burning things in order to make heat, in order to make energy, and that's really what we do. And of course, we need oxygen in order to do those things. The problem is, cells inside a three-dimensional structure, like the body, are always getting different amounts of oxygen, and it can depend on different levels of blood flow. It can depend on the fact that the tissue itself might be using a lot of oxygen at any given time. My brain is probably using a fair amount right now, my heart, because it's probably beating a bit fast. And so that's maybe got sort of, it's using more oxygen than if I'm lying down and sleeping. And so because the cell has a very tightly regulated little furnace in it, it has to adjust to these different levels of oxygen in a very fine-tuned way. And if it does this badly, it can even be fatal for the cell. So this is really, the prize is for this sort of almost like a rheostat or a thermostat for the oxygen levels, or a damper that you'd have on your furnace to let in more or less oxygen at any given time. So it's just right, so the flame burns just right.
And if we take a small step outside the cell, how does this discovery affect people's lives?
Well, it affects people's lives because it really will help us and is already helping people develop new medicines. So basically finding different ways to influence this fundamental process has already shown itself to be potentially very useful in very clear medical applications. So as I mentioned in my presentation, for example, if you want to increase the levels of your red blood cells, this factor comes into play. And that, of course, makes sense because if you're at high altitude, you want more red blood cells to help you carry oxygen. If you have an accident and you lose a bunch of blood, now there's not enough red blood cells to carry the oxygen. So you want a signal that tells your body make more red blood cells. And this is a fundamental part of that signal. It's the cellular aspect of it. So a drug that can raise the levels of this can trigger new red blood cell production. And in fact, such a drug, as I mentioned, has already been approved in China and is under consideration in the Western countries as well, including the EU and Sweden.
So in order to describe this prize in an accessible way, what kind of metaphor would you use?
I'd say, I of course would go back to the candle or the furnace and say this is a way for the cell to adjust how much oxygen or adjust its response to the amount of oxygen it gets, because of the cell directly can't change how much oxygen it gets oftentimes. But it's an interesting prize in that that's of course at the cellular level, but it also controls much more far-ranging things. So you can think of a cell having to change its metabolism. Okay, that's at a very basic level, but the cell also, when it senses it doesn't have enough oxygen, triggers, for example, the production of new blood vessels. So if I get a cut, those cells now don't get a good supply of oxygen because I cut the blood vessels that are taking blood there. So the cells now release factors that induce new blood vessel formation, also through this factor. Or, again, if I go to high altitude, now I'm not breathing in enough oxygen, and now my body sends a signal saying, make more red blood cells. So it ranges from the cell all the way up to the body, basically.
So if we turn to the laureates, William Kaelin, Sir Peter Ratcliffe, and Greg L. Semenza, what have they done?
What have they done
in this discovery?
In this discovery, well, I know them all. They're all very fine fellows, and they all did very essential parts of this process. Semenza, Greg's initial and very important contribution was first to sort of help define the region of a gene that is basically allowing this to happen, and then it was found that this region was found in lots and lots of other genes. Peter Ratcliffe also participated in that process of defining that. Then Greg actually was the one who cloned and isolated this factor called HIF, and he was the one who named it. And so that has really been the foundation for this discovery in many ways. Peter Ratcliffe came in and made an association between this tumor suppressor gene called von Hippel-Lindau, or VHL, and the regulation of this HIF gene, and that's really an important part of how HIF senses oxygen. And Bill Kalin came in and also made that association, and he and Ratcliffe helped to define the switch, the oxygen-dependent switch, really the knob on the dial, really, that ultimately helps you understand how all of this works. So all three were necessary components, really, or actors in figuring out how this whole thing works. Although they haven't collaborated with each other, they've all been closely associated with the field through the course of all of this.
And how come the prize is awarded now? I mean, some of those findings were in the mid-90s. What has happened to make it sort of such an actual thing?
I can't speak to real specifics because, of course, some of that is confidential in terms of the deliberations of the assembly. But it's very clear that we now understand this fundamental biological switch that really impacts all our lives as living creatures here on Earth breathing oxygen. It's really one of the most important things our cells know how to do. In fact, if an embryo doesn't have this HIF gene, it won't survive past very early embryogenesis. So even in the womb, our bodies need this gene in order to do everything they do. So I guess you could say a direct answer to your question is it seems like a complete and clear story.
And the laureates, I understand the Nobel Committee has been talking to them. So what were their reaction?
Well, of course, that's Thomas, the secretary, is the one who calls them on the phone. But from what I understood, they were all very happy, as one should be, and very excited. And I certainly, we're looking forward to welcoming them to both Stockholm and Sweden in December.
And finally, if you have 30 seconds to explain the main impact of this discovery in an exciting way, what would you say?
I'd say this is what scientists often toss around, this phrase, textbook discovery. I'd say this is really essentially a textbook discovery. This is something that basic biology students will be learning about when they study at age 12 or 13 or younger biology and learn the fundamental ways in which cells work. This is a basic aspect of how a cell works. And I think from that standpoint alone, it's a very exciting thing.
Thank you very much.